turbo engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
questions,
Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists, the programme where we bring you the latest breakthroughs in science, technology and medicine. I'm Chris Smith. In the programme this week, a rundown on the week's Science Nobel Prizes, why paracetamol might cause behavioural problems in children born to mums who use the drug when they're pregnant, and the spacecraft that crashed into an asteroid on purpose.
The Naked Scientists podcast is powered by ukfast.co.uk.
Now, this week's show falls during a very special week in the annual scientific calendar. It is, of course, Nobel Prize Week, the time of the year when the world celebrates the very best in research and discovery. And we wanted to pay tribute to the work of the fantastic scientists who've won them by explaining a little bit about each of the prizes that's been won so far. The Physics Prize has gone for a subject that Einstein himself was sceptical about and called spooky action at a distance. James Titko.
This year, the Nobel Prize for Physics was awarded to Alain Aspect, John Clauser, and Anton Zeilinger. The official citation sets out the awarding of the prize for experiments with entangled photons, establishing the violation of Bell inequalities and pioneering quantum information science. So let's unpack this. Quantum theory is the branch of science that aims to continue the work of some of the most brilliant minds to have ever existed, those whose imaginations were captivated by the great cosmic questions, from Newton to Einstein to Hawking. It is the next frontier for scientists seeking to understand the fabric of the universe. Yet despite the very big questions it endeavours to answer, quantum mechanics describes the behaviour of systems at the atomic scale and smaller. In the quantum world, matter has wave-like properties, in contrast to the more rigid and tangible view of the world posited by classical physics, that's objects and phenomena we can observe with the naked eye, as set out by Isaac Newton. Where our Nobel Prize winners come in is in their work on a smaller aspect of the overall field of quantum mechanics, the branch called quantum entanglement. This is the idea that two particles, even separated by considerable distance, remain in touch with one another and even dictate the properties of the other. One way to think about this is to imagine being given one of two balls. You are told that one of the balls is white and the other is black. So, by deduction, if you receive a white ball, you know the other ball must be black. But where this becomes interesting is that, in the quantum realm, until one of these particles or balls is examined, the properties of the other are not determined. So both of them are effectively grey until one of them is looked at. At that point, one turns white and the other black. Mind-boggling though this sounds, Aspect, Clauser and Zeilinger conducted the experiments to show that this phenomenon, theorised in the 1960s by Northern Irish physicist John Stuart Bell, is real. How it works, we still have no idea. Even Einstein called it spooky action at a distance. Nevertheless, quantum entanglement could have many applications for use in the modern world, in quantum computing to help solve complex problems, and in encryption, useful for communications and banking. Despite this breakthrough, there is much work to still be done in the field. From a theoretical point of view, a new Einstein is needed to blow our understanding of the nature of matter wide open. Progress like this, however, indicates that we're on the right path.
James Titko reporting on this year's Nobel Prize for Physics, and we'll be looking at the prizes in chemistry and physiology or medicine later in the programme. Paracetamol is the world's most popular painkiller. It's a crown it stole from Bayer's aspirin, the world's first trademark drug, on safety and side effect grounds. It's also widely used and regarded as safe in pregnancy. But Penn State University's Christine Schneider has found a possible association between paracetamol use during a pregnancy and subsequent behavioural and sleep problems in children by the age of 3. There was a 20% increase in the risk of that happening.
We had seen some literature coming out showing associations between
using paracetamol or acetaminophen during pregnancy and problems with neurobehavioral child development.
And you wanted to find out whether it was a causal thing. Does using this agent cause this to happen, or might there be other factors that actually account for it?
Absolutely, that's exactly what we were looking for. So one of the great things about our study, I think one of the strengths is that it followed women from starting during their pregnancy through 3 years postpartum. So we were able to kind of see what came first. And we know then we had that they're taking acetaminophen or paracetamol during pregnancy and to follow them to see how their children were at 3 years.
How many women did you follow up?
We followed up over 2,000 women.
And what were you asking them? Did you or did you not take paracetamol, presumably? But how fine-grained was the information?
In their last trimester of pregnancy, we asked everyone in the study what medications they were taking during their pregnancy, including vitamins, also why they were taking the medication. So we were able to then look through the medications that they listed for each woman and see which medications had paracetamol in them, or if they were just taking paracetamol alone.
Did you get some insights into how much they were using? So would someone tick the same box if they had one headache once and took one paracetamol versus someone who had really bad joint pain all through pregnancy and used it every week?
We tried to get at that, but the data weren't very reliable. We actually have a new study underway that we're hoping to get better at the dosage and frequency of acetaminophen.
And when you crunched all the numbers, what was the relationship that emerged?
We found an association between paracetamol use during pregnancy and attention problems and problems with sleep among children at 3 years old.
Was the effect strong, or are we talking about it's just in there, just above chance?
It's 1.2 times more likely. So I wouldn't say it's an extremely strong association, but the association was statistically significant.
And any clues as to who's vulnerable? Was this across the board? Or if you then begin to ask, are there certain groups in there who mum takes a lot of paracetamol, perhaps because she's got some other underlying condition and it's the other underlying condition that's causing this? Or were there other factors that could be discounted and you could say, no, look, this is purely because of exposure to paracetamol?
I think one of the strengths of the study is that we were able to take into account those factors you mentioned. So because we asked why they took paracetamol or acetaminophen during pregnancy, we could then take that into account, and we found this effect of acetaminophen as an independent effect. So it wasn't a fever or infection.
The other thing that is an important component of proving that something causes something else is it's got to be biologically plausible, whereby you can say, well, we know how this thing affects the body, and if that happens, we can explain how that might translate into this outcome. So can you link a pregnant woman taking paracetamol and a child with neurobehavioral problems?
Yeah, there has been research that shows paracetamol may disrupt cell development. So we think paracetamol could disrupt cell development and also result in placental damage.
When did paracetamol first begin to be used? And when were we told it's quite safe to use this in pregnancy? Because it seems to me that we've got very good at diagnosing things like developmental disorders and behavioural problems in the modern era. There seems to be a lot more of it around now. Is that because we're better at diagnosing it, we're picking it up more, but it was always there? Or is there some kind of coincident onset of we're seeing more of these cases because there were more mums using paracetamol when they were pregnant?
That's a fabulous question. I think we're really trying to disentangle that. I'm really not sure if we know the answer to that. I think we are getting better at diagnosing developmental delays, but I do think that we're still trying to figure out, and the research is kind of showing more and more, there could be several factors related to behavioral problems in childhood, genetics, and environmental exposures, possibly in utero. So we're really working to figure that out.
It's, as you say, critical, though, that we do, isn't it? Because this is one of the world's most popular painkillers, regarded as one of the safest and very broadly used during pregnancy.
Absolutely. This is something that really needs more focused research. Literature is emerging. There has been a call to action to reduce paracetamol use during pregnancy that came out last year. And I think we're really showing these trends that people should be a little more cautious as they consider using paracetamol during pregnancy.
I do feel for pregnant women, though, because they're frightened to do anything these days, aren't they? What should then a pregnant woman listening to this do if she has a headache?
I think she should talk to her doctor to weigh the options and what she could do, or maybe talk about what medications are safe with her clinician, and maybe bring up some of these findings that we're talking about to see what her clinician thinks.
I think
That's
there's going
what I
to be
would
a lot of
recommend.
general practitioners are going to be confronted by patients walking in there with your paper. And they're going to say, what do we do?
Well, maybe so. It's a risk benefit that people need to take into account.
Kristin Schneider, and that work was just published in the journal PLOS ONE. Well, now it's the turn of chemistry to go under our Nobel microscope, and to explain who's won this year's prize, here's Risa Bagwandin.
This year's Nobel Prize in Chemistry has been awarded to Carolyn Bertozzi, Morton Meldal, and Barry Sharpless for their pioneering work in the field of click chemistry. Conceptually, click chemistry is a bit like molecular Lego. It's a way of stably snapping together the building blocks of molecules to efficiently construct bigger, more complex chemicals. Imagine trying to assemble a complex piece of flat pack from IKEA. You have all the pieces and know what the end result should look like, but you don't have enough pairs of hands to stop the thing wobbling while you add the next component. The result is that it falls apart faster than you can make it. Click chemistry basically solves this problem by handing the builder a bunch of cable ties to hold the right bits of his molecule together so it doesn't fall apart during construction. The result is, broadly, if we know what molecule we want to make, now we can use click chemistry to create it, but without the lengthy reactions, wrong turns, or low efficiency of the past. From chemical manufacturing and engineering to the pharmaceutical industry, the potential of this technique is huge. And because the technique can be water-based and made to work without resorting to harmful conditions or substances, it can even be applied inside living cells. This was a breakthrough demonstrated by Carolyn Bertozzi. She added a click chemical to cultured cells, which incorporated them into sugars they internally produce. This enabled Bertozzi to hitch up colored dyes to track how sugar-containing molecules are moved and processed, and even how whole cells move and migrate. This has subsequently helped to reveal how cancer cells evade our immune systems and spread, and led to techniques that can target radiotherapy directly to tumors, meaning healthy cells nearby are less likely to be harmed. Bertozzi said she was absolutely stunned to get the call from Stockholm. Sharpless, though, is taking it in his stride. He's won the Chemistry Nobel Prize once already. For him, presumably, chemistry just clicks.
Thanks very much to Cape Town chemist Risa Bagwandin, and we'll be looking at the Nobel Prize for Physiology or Medicine in a few minutes' time. Meanwhile, a new way to break down and recycle plastic has been announced by scientists in the US this week. This is very welcome news since the world produces millions of tonnes of plastic every year and less than 20% of it gets recycled. Most of it ends up in landfill, some gets burnt, and a significant amount ends up in the ocean, where it degrades into potentially harmful microplastics. Part of the problem with plastic is that it's an unnatural chemical which nature has no easy way to attack, so it just hangs around. But John Hartwig and his colleagues have developed a relatively low-temperature chemical process that can break into the long chains of carbon atoms that make up polythene, or polyethylene as it's also known, and break them into shorter molecules of the gas propene, which is a highly valuable raw material for making many other materials. He told me how it works.
Polyethylene is multiple units of ethylene put together through carbon-carbon bonds, which are very stable bonds. So what we've done is a three-step process where first we introduce a change to the chain that provides kind of an Achilles heel, where we can then cleave that chain. And maybe a good analogy is if you think about the polymer chain being actually a physical chain like you'd buy at the hardware store that has these links that are very stable and you can't pull them apart. But if we make a chemical change to our polymer chain that transforms one of the linkages to, say, a clasp like you'd have on a necklace that could then be opened, now we have a way to break apart those chains under milder conditions. And then there's two additional steps to rearrange the chains and attach a very small molecule to break those down into very, very small pieces.
I was going to ask how many of these so-called clasps one would need to insert in order to get this thing to fall apart, and under what sort of conditions do you need to do this? Because one of the criticisms of recycling processes is that if you're not careful, you end up with a bigger carbon footprint than the one you're saving.
Right. Fewer than 1% of those chains need to be transformed into a clasp. That transformation of the chain to the clasp, we do that at 200 degrees, which is pretty low temperature for chemical processes.
And how do you actually do this? What is the mechanism by which you change those chains so that you can then do this modification and make them fall apart?
The first step is a reaction called dehydrogenation, where we remove hydrogen, therefore the name. We do that with a catalyst. The catalyst has platinum in it. It's a platinum zinc catalyst. That's the best one we've found. And then once that chain link has been turned into a clasp, we use two catalysts. One that is a subject of a Nobel Prize in 2005, olefin metathesis catalyst, it's called. And so that basically would take two chains that would have a clasp in it, undo the clasp, and put the chains back together. And then we have another reaction that would take that clasp. We walk that clasp in just a little bit into the chain and then break the chain at that point to make a small piece off the end and a shorter chain. And then we do that about 1,000 times to turn the solid polyethylene into a gas, propene. That's our final product.
Is this a single reaction that you can do those three reactions in one place, or do you have to do one thing, feed it into the next, and then feed it into the next? How practical is this?
Right. We actually run it as two. We run the dehydrogenation first, and then this moving of the clasp and opening and closing of it, those two reactions are run at the same time. There's also a very closely related work that has just appeared also in publication, and they run it all together, but the yields are lower than in our case. So in principle, all three steps could be run together. In practice, our highest yields come when we run them separately.
And what sorts of yields can you get? Is this a practical and viable, as in industry-ready technique? Could we see this digesting plastic bags anytime soon?
Well, the answer to the last question is it will take a lot of time to develop. But the answer to the first question about the yields and whether it's practical, the yields are very high, actually. We get almost 90% gets converted into propene. And even on waste plastic, the yields are around 50%. And they're probably higher because those plastics aren't all polyethylene. They contain fillers and color, and dyes and various other additives to them. But it takes decades between the time of, say, a first paper like this and when it becomes practical. So we need to have the catalyst be very, very stable to be able to run for a long period of time, make many molecules of the product, maybe 100,000 times. And right now we're at maybe 100 instead of 100,000. So we have a lot of development to do, but we hope this provides motivation to do the work and demonstration that it could become feasible in the future.
The problem plastic has at the moment, apart from it is a wonderful material when we first make it and first use it, it becomes a pain when we get rid of it. It's valueless almost, isn't it, when it's a waste material? Does your process mean that potentially we return value to waste plastic, and does that solve our problem in the sense that it incentivizes people to pick it up?
Well, that's exactly the idea. So what we want to do is to take chains back apart to the small molecules like propene that has only 3 carbon atoms in it and is a feedstock chemical to make all sorts of other things. That's the idea of this kind of recycling we call chemical recycling or advanced recycling.
Some good news. John Hartwig, he's at UC Berkeley, and the work that we were discussing has just come out in the journal Science. Well, it's time now for another Nobel, and this one is the prize for physiology or medicine. And it goes to a researcher who's pioneered a field of science that's revolutionised our understanding of life, both present, but also life long gone. We'll tingle.
This year's Nobel Prize for Medicine and Physiology goes to Svante Pääbo, a Swedish geneticist, for completing the seemingly impossible task of sequencing the genome of our extinct relatives, the Neanderthals. But why was it deemed seemingly impossible? Well, for around the last 30,000 years, Homo sapiens, which is to say us, have been the only species of hominid left on the planet, meaning any samples of Neanderthal DNA, such as bones, would have undergone a lot of degradation. DNA is a resilient molecule, but just like any other, will fall apart given enough time, which meant that any useful material left in 100,000-year-old samples would have been sparse at best. This also doesn't even begin to cover the amount of bacterial contamination that the bones would have gone through in the past few millennia. Recreating the Neanderthal genome, then, would be the equivalent of trying to do a billion-piece jigsaw with a blurry reference picture whilst most of the pieces are nearly identical. Small wonder, then, that the process took the best part of 20 years. But, even against such fearful odds, he managed it. Along the way, he created and refined several new techniques of repairing and sequencing DNA. He designed clean rooms, dedicated to handling ancient DNA, which protected his fossils from being contaminated by living humans. And advances in sequencing technology, aided in no small part by the Human Genome Project, meant he could decipher the DNA found in ancient bones. The findings of his work show that up until 40,000 years ago, we were frequently mixing with Neanderthals. Even after 1,400 generations, up to 4% of our DNA can be traced back to them. In his own words, they still contribute to what we are today. The knowledge of our genetic makeup can have massive health implications. In fact, a paper co-authored by Pabo himself concluded that the coronavirus caused more severe symptoms in people who had inherited a segment of Neanderthal DNA. Knowing what we're made up of means we can more effectively understand how best to treat illnesses that have strong ties to our genes. But by contrast, knowing what parts of us come from our ancestors can also show us what parts are unique to Homo sapiens, and perhaps highlight what allowed us to become the success story we are today. In the words of Johannes Krauss, a paleogeneticist at the Max Planck Institute for Evolutionary Anthropology, we've never come so close to understanding what makes humans human. Make no bones about it then, Svante Pääbo is the deserved winner of this year's Nobel Prize for Medicine and Physiology.
Will Tingle on this year's Nobel Prize for Physiology or Medicine.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
You're listening to The Naked Scientists with me, Chris Smith. Still to come, the arms war that's raging on the streets of Sydney between bin owners and local birds. Before that, though, it's made a mint for Hollywood, but the idea of nuking an incoming asteroid to avoid an Earth impact that might otherwise wipe us all out, is that more rooted in reality than science fiction? Well, last week, NASA's DART mission slammed an impactor weighing 0.5 ton into a tiny moon orbiting an asteroid to see how practical the approach might really be. They estimate, long-term, that the collision might move that moon about 1% closer to the asteroid. Well, now the dust has settled, Open University planetary geologist David Rothery has been taking a look at the aftermath and the rationale for doing it for us.
DART was launched a year ago to what's known as a near-Earth asteroid called Didymos, which has an even smaller moon. The moon's called Dimorphos. It's 170 meters in diameter. Didymos is about 800 meters diameter. It was deliberately crashed into the moon of Didymos, so crashed into Dimorphos at just over 6 kilometers a second to see what change could be made to the orbit of Dimorphos. The point is, in the future, we might want to change an asteroid's trajectory if it's heading towards the Earth. So this was an asteroid redirection test tried out on the moon of an asteroid, because when you've got a moon in a regular orbit, it's easy to see what's happened to the trajectory. You can just measure the orbital period of a moon and see how that's changed by the impact. And there was this almighty collision. And the plume thrown up from this collision was seen by telescopes tracking it from the Earth, and also seen by a little CubeSat, which the DART probe released a few days before impact, which was filming from space and saw the impact as well. And the way the ejector has been flung out is intriguing people. It's come out in streamers rather than a continuous cone. So that's interesting. But the main science that's going to come from this is, how has this asteroid moon been disturbed in its trajectory?
We don't think, though, that there are any objects which are on an earthbound collision course at the moment, do we? I mean, just to kind of calm people down, this is not because NASA knows something.
But there are no dinosaur killers lurking out there. There's nothing that's going to disturb global climate for a decade. But If one of these things hits the ground in a city, it will destroy that city. So you don't want it to happen. So we do want to know how to deflect these things away if we know that there's one coming. And the idea would be to get it several years, several orbital passes before it's going to hit the Earth, because you only need then to disturb the trajectory just a little bit so it misses rather than hitting. So the longer in advance we can do this, the better. But what we weren't sure is if you slam into an asteroid, is it going to fall apart? The surface of Dimorphos is just made up of a massive interlocking, very large boulders, and I guess smaller stuff in between. Now, it's a rubble pile. Was it going to fly apart, or was it going to stick together and the whole thing be disturbed its trajectory as a single mass? It seems that the latter has happened, which is good news. It wasn't blasted into smithereens.
Because that matters, doesn't it? Because otherwise you turn one threat into 1,000 threats.
Absolutely, yeah. And it's good that that doesn't seem to have happened in this case.
Are they going to go back and have another look, or will all of the observations now be from Earth?
There are still data coming back from Larcia Cube, which is the Italian CubeSat that was deployed by DART itself. But there is a follow-up mission as well. It's called HERA. It's a European Space Agency mission, which will launch in 2024, and 2 years later will arrive. It will be able to see the crater formed on Dimorphos by the impact. And I guess it may refine the orbital period and the shape of the orbit of Dimorphos around Didymos, but we will have that from ground-based observations as well. By measuring the orbital period, which we can tell from the Earth, we know how much the Moon's trajectory has been disturbed by this impact. But the follow-up from HERA will add another layer of information into that. So it's going to be a well-studied system. They're both interesting shapes. They're both made of rubble. So in their own right, they're interesting objects as well.
And does the planetary geologist in you want to know more about what these objects were made of? Because obviously doing things like this does expose to us what they're made of. And they're intriguing things because they date from the very same material that formed the solar system 4.5 billion years ago.
Yes, I don't know how primitive this object is in terms of what processing has gone on. Some of these asteroids have been damp in the past and water has migrated through them and altered the minerals. It's too faint probably to have a very good spectrum of it. I'm speaking outside my sphere of knowledge now, but looking at the pictures, the jagged rocks of all sizes on the surface that probably give it quite a bit of cohesive strength intrigue me. What's gone on to process this material so it's just a rubble pile? And yet it's a pile of rubble which seems to have hung together pretty well under this sudden impact. If you hit this thing slowly, it might break apart. If you hit it fast, the shock of the impact will tend to make the interlocking pieces hold together better. So if you want to deflect an asteroid, you've got to do it at the right speed as well. So there's a lot to learn about how to deflect asteroids. This was just the first well-controlled step towards doing that.
Thanks very much to David Rothery. The evolution of animal limbs is a subject that's perplexed paleontologists for a very long time. Fossilisation is regrettably a very rare occurrence. Fewer than 0.1% of all species over time that have lived have ever become fossilised, and so finding missing links in evolutionary biology is a challenge. This was very much the case for the origins of limb development, at least until very recently. Galeospider are an extinct group of jawless fish that date back about 430 million years, but until recently only their heads had been preserved. Will Tingle spoke with Phil Donoghue from the University of Bristol about how this new Galeospid discovery, which sheds light on the moment when these early fish developed the paired fins that may ultimately have led to our own arms and legs.
The real frustration, despite having tens of thousands of these fossils, is that all we've had until now are their heads. And the discovery that we've made now is a series of specimens of Galeospids which preserve the rest of the body, providing us with insights into how the rest of the body plan of jawed vertebrates evolved.
From what I've seen of it, it is a striking looking specimen, this fossil. But for the benefit of our audio listeners, what does it look like?
Well, it's a beautiful fossil, but beauty is in the eye of the beholder. It's basically a dead fish. and is preserved, at least as we see it, with the bottom of the body facing upwards. And it's fairly squashed, but the amazing thing is that it has a series of paired fins that extend vertically, almost entirely three-dimensionally, extending up from the body. It's the kind of preservation you never see in fossils of this type. And these fins extend all the way from the back of the head, all the way to the tip of the tail in a way that we don't see in any living vertebrate, and indeed any fossil vertebrate until now.
And how did you come about this fossil?
Luckily, I had a graduate student about a decade ago with whom I started working on Galiaspids. He's since gone back to China, and he and his colleagues made these amazing discoveries and were generous enough to invite me in to help describe them and interpret them.
When you saw this, what gave it away to you that this fossil, this Galiaspid, was the one that you were looking for?
Well, it was easy, really. It wasn't just a head. There was another 3/4 of a body extending from the back of the head. And it had anatomical features which we really didn't know one would have guessed that Galiaspids possessed. And the key thing is that these fins, really, all jawed vertebrates have a pair, unless they've lost them, a pair of anterior appendages, whether they're fins or whether they're limbs, and a pair of posterior appendages, again, whether they're fins or whether they're limbs, like our legs. And they're entirely separate from each other. But in this Galiaspid, which is probably the first vertebrate to have paired appendages, they extend all the way, as I say, from the back of the skull all the way through to the tip of the tail. And what this suggests is that this is the precondition from which separate fins and limbs ultimately evolved.
And whilst this might sound like a bit of an obvious question, why do you think they evolved the first limbs? What was the evolutionary advantage to it?
Well, the evolutionary advantage seems to have been if you extend from a cylindrical body, if you extend paired structures out, they appear to just generate lift. They allow you to effectively swim without putting any energy into the system whatsoever. It's just a product of the geometry of your body. The fins that these organisms possessed weren't muscular. They couldn't move in any way like the fins of all living vertebrates and limbs of all living vertebrates. That's a phenomenon that evolved seemingly much later in invertebrate evolution. But nevertheless, it's this precondition, this adaptive benefit of being able to locomote without expending any energy seems to be the reason for why paired appendages first evolved.
And how does this discovery change our preconceived timeline of appendage development?
So previously, again, based on fossils, if you set aside our new evidence from Galiaspids, it seems quite clear that a more advanced group called the osteostracans, they just have a seemingly just paired equivalents of our arms. So a pair of anterior fins, and these were flexible and these were muscular. And so we don't really see the equivalent of pelvic appendages, the equivalents of our legs, until the origin of jawed vertebrates. So all of the evidence coming from developmental biology, trying to understand the developmental basis of how fins and limbs evolved, has been interpreted within this framework from the fossil record, suggesting that pectoral fins evolved first and pelvics evolved later. But the evidence from Galiaspids suggests that actually, no, that's not the case. These are just peculiar conditions of derived osteostracans, for instance. And the condition in Galiaspids suggests that the primitive state is one in which you have continuous fins extending from a pectoral through the pelvic position all the way to the tip of the tail. And it's actually the separation of that continuous zone of competence for developing fins that the differentiated anterior and posterior paired fins ultimately evolved.
Phil Donoghue, and that paper was just published in the journal Nature. Cambridge scientists have discovered something surprising going on in all of our cells that we'd overlooked for years. Structures called mitochondria, which used to be bacteria before they merged with our ancestors' cells billions of years ago in a partnership that now supplies us with the energy that keeps us alive, contain their own bacteria-like DNA that's separate from our main cellular genome. But Patrick Chinnery has discovered that pieces of this mitochondrial genetic code periodically get cut and pasted into our own chromosomes. And this is especially true in cancers, which might explain some of the growth and resilience characteristics that cancers display.
We got the first clue a few years ago when a group in the United States reported the transmission of mitochondrial DNA from fathers to children. This was a crazy suggestion to the field, so we went looking for an alternative explanation. And what we found was that it wasn't the mitochondrial DNA that was coming from the father, it was the nuclear DNA. But what had happened is bits of the mitochondrial DNA had gone into the father's nuclear DNA.
We should clarify that the mitochondria, which are these cell powerhouses, are in the cell, therefore they're in the egg cell that gives rise to an individual. They're not in the sperm. So when the sperm, as in they're not transmitted from the sperm, which is why you're saying there's that distinction. You get your mitochondria from your mum, not from your dad.
All from our mum. They thought it was coming from the dad. We've shown it's not the case.
How did you prove that that was what was going on?
So we worked with colleagues at Genomics England who've been carrying out the 100,000 Genomes project. People across the whole of the NHS have been contributing samples to this, and we looked at the genomic sequence of over 60,000 individuals and 12,000 cancers and looked for the signature of these bits of mitochondrial DNA across all of these individuals.
How is it getting from the mitochondria, which has got its own little circle of DNA inside these structures inside our cells, and it's getting from a totally different part of the cell into the nucleus, the headquarters of the cell, and into a chromosome in there? How is that happening?
It's a very good question, and we don't know the answer. We're embarking on a programme to work that out. We think what's happening is that as the mitochondria are recycled, bits leak out and cross across into the nucleus through holes in the membrane that surrounds the nucleus itself and integrates them into the chromosomes, which is how the nuclear genome is packaged.
The mitochondrial DNA has got instructions in it that keep those cellular powerhouses happy. It's how they operate. What's the consequence of pasting bits of those genetic instructions into the main chromosomes in our cell? Is there one?
Well, it all goes back to how mitochondria originated. And the idea was when they first came into the cell, they passed on certain functions to the cell. And to do that, they passed DNA. So actually what we're seeing now is a consequence of that process. All of this was thought to happen billions of years ago, but actually we've measured it happening in families, and 1 in 4,000 families, a new bit of mitochondrial DNA goes into the child. It's never been seen before at that rate, and in cancers, it's even faster.
What about the reverse direction?
So it doesn't happen, and there are several reasons why that might be the case. One is that you've got many, many more copies of mitochondrial DNA in each cell than the nucleus. That's one reason. The other is that there are holes in the nucleus that allow mitochondrial DNA to go in, but not the other way around.
Right, okay, so it is a one-way street. And the consequences of this stuff going in, you mentioned cancer, and it does appear to occur more frequently in cancer cells when you look. Now, is that just a hallmark of the fact that cancer is a genetic disease, and therefore a genetically unstable cell is more susceptible to this happening, or is there more to it than that?
We've looked at where these stick in the nuclear genome and looked at the pattern of that. And what we found is that they sit nearby breaks in the genetic code. So anything that causes your genetic code to break up will attract these bits of mitochondrial DNA that effectively behave like band-aids in the short term to repair the genetic code. So in cancer, you get a very unstable nuclear genetic code, and one consequence of that is the mitochondrial DNA can find its way in there.
Patrick Chinnery, and he just published that work in the journal Nature. The suburbs of Sydney are playing host to an unlikely battle of wits. Residents are putting their bins out for collection and have found themselves confronted by hungry cockatoos eager to open up the bins and help themselves to all the leftovers. To deter the bin raiding, locals are resorting to increasingly complicated ways to safeguard their trash. But the indefatigable cockatoos are just working out how to get round them and then teaching each other the trick into the bargain. Now, cockatoos are one of the most intelligent bird species, and they can copy an action after they've seen it only once. Indeed, their bin raiding antics have spread through the suburbs so quickly that now commercially available cockatoo locks are doing a roaring retail trade. Will Tingle spoke with Barbara Klump from the Max Planck Institute of Animal Behaviour, who's been following this arms race on both sides. People sharing defence ideas and birds sharing attack plans.
Over time, more people protect their bins, but also more people escalate the method they're using from more low-level protection to higher levels of protection. This was in direct response to the cockatoos solving some of the methods. So we think that this might be the beginning of what we call like an innovations arms race, where one species changes the behavior in response to what the other species is doing.
There have been cases of arms races between two species in the natural world. You've got your bats and your moths, you've got your newts and your garter snakes. But these cases are more arms race based on evolutionary adaptations and physiological changes. So is this the first case that we've seen of an arms race based on behavioural changes?
So I don't know whether it's the first case of a behavioral arms race, but it's certainly the best documented, I would say, because when we looked through the literature, we were actually quite surprised how little there was. So there's mention that it might be happening between killer whales and their prey, and then of course lots of like anecdotal observations where people have told me they have like their personal arms races with the squirrels that try to get into their bird feeders. There is definitely this interaction happening, but I think it hasn't been studied in detail so far. But I think it would be very interesting to study this in other species as well, because as you say, most arms races that we know of are on these like really large evolutionary timescales. But with like behavioral or innovations arms races, this happens much, much faster. And that allows us to also like study it in much more detail. And I think it could be a really, really fruitful like future research avenue.
Do you see the amount of arms races that humans are going to be involved in terms of wildlife interactions changing in the future?
I could imagine that it will increase just because cities expand and we will share more and more of our space with wild animals. So that naturally also increases the chances for human-wildlife conflict, but it also increases the chances where such arms races can develop and then can be observed and studied. So I would imagine that it will increase in the future.
And how do you see this one ending? Do you think cockatoos are going to win, or do you think humans have managed to lock them out?
That's a very interesting question, and I don't know. So currently we know that the cockatoos can open some, but not all of the protection devices that people have come up with. But that doesn't mean that they won't learn to defeat them in the future. And I think there's so many other factors that play into this arms race as well that, make it quite difficult to make a prediction. But I can tell you that I'm super excited to find out in the future.
Barbara Klump, and that paper is in the journal Current Biology. Well, that's it for this week. The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education. It's supported by Rolls-Royce. I'm Chris Smith. Thank you for listening, and until next time, goodbye.